Hi guys, welcome to our second unit, which is animal body plans. For this unit, we are going to be going through a lot of information, and so please feel free to pause the video and write down any questions that you have. There are a lot of terms, and although we'll have all the definitions for the terms available to you for your next assessment, it is important that you understand how it's all connected. So make sure you have a good clarity understanding of all of this. For this unit, you need to make sure you understand this drawing and you understand what the gastrula, endoderm, mesoderm, and ectoderm from the last class was. So pause the video now, label this picture. If you are not able to do that, you need to go back and review the information. This is an important aspect of this lesson today. Okay, so we're going to start talking about animal classification. And when we talk about animal classification, we are organizing our animal classification, but something we call our body plan. So the body plan is a set of developmental features or common physical characteristics that a group has that divides them into their own group. So you notice here there are this is our cladogram for animals, and there are 35 different phyla that animals get classified. We will not be covering all 35. We're going to cover the majority of the ones that you see here. One thing that I want to remind you is that here's your ancestor. These are going from least complex to most complex. The other thing that I want you to notice is that every time there's a branch, this branch continues on to become more and more complex. So that's another way to look at it. Okay. Our first unit of division, our first plan, is the tissues. And for the tissues, there's either tissues present or tissues not present. Remember, a tissue is a group of cells that work together to accomplish a task. So you may have your vascular tissue. You may, for example, in plants that help carry materials. So here we have no tissues, and the only thing that organism that does not have tissues, like I mentioned the last time, is sponges. Everything else has tissues. The next way in which we divide or characterize animals is by their symmetry. Now, there are three different types of symmetry. Here we are showing two in our cladogram, but in reality, there are three. Symmetry is how a body is structured or how symmetrical it is. It influences how well the body moves. So, in other words, the more complex the symmetry, the better the movement. The first type of symmetry is asymmetric. So what does that mean? We look at this organism, and when we look at this organism, we can divide him in sections. We try to divide him here, try to divide him here, try to divide him here, and you cannot put a plane that splits this organism into equal parts. So they're considered no symmetry, not symmetrical, and so therefore it is asymmetrical. This is the most primitive type of organism that you would find. The next type of symmetry, so this is more evolutionized than asymmetrical, is radial symmetry. So these organisms are radially symmetrical. So if I take and try to split my sea star, and here I'm going to have two equal parts. In here, I'm going to have two equal parts. In here, I'm going to have two equal parts. Same thing with this organism here. No matter what we divide him, and we can divide him multiple ways, and we're still going to have equal parts. Just like an example that you have in your house is your bicycle tire. You can split the bicycle tire in multiple ways, and they're still going to be symmetrical, meaning the two sides are going to be equally the same. So they have multiple. Radial symmetry means that there's multiple ways that it can be divided and have symmetry. The last one is bilateral symmetry. Bilateral symmetry is when you take an organism and you can only divide them in half one way. And so that's like humans. Humans are bilateral symmetry here. In other words, I can't split the human like this because here I have feet and here I have a head. So they're not equally, but these are all bilateral symmetry. What does that mean? There's a lot more movement that these organisms are able to do. How? You have hands, you can touch, you can respond, you can turn around and run away and follow something. When you have an organism like this, they have sensory all around. So they have radial sensory, they can feel all around it. So if it feels something here and it feels something here, it's going to feel it. But it doesn't have a centralized location that it can chase or follow an organism or follow the light. One of the things that bilateral symmetry brought is that it allows us to label different sections based on the symmetry. So anything above the head is called anterior. Anything on the bottom is called posterior. Anything on the back is called dorsal. And if you think about the dorsal fin of a fish, it's the one that goes in the back. And anything on the belly is called ventral symmetry. Another benefit of bilateral symmetry is that it brought this incredible evolutionary trend, which is called cephalization. Cephalization is a development, essentially the development of the brain. So the nervous system, sensory organs, and a brain that can process this information. Now, the level of the head and the level of brain, so cepha means head, but the brain development varies in species, but bilateral symmetry allows these organisms to have a head and to have sensory organs that they can chase and they can go after. Our next group is called body cavities. And so I'm going to give you some new terms here. So now we are realized this, that, we're done with radial. So all the bilateral symmetry organisms have a body cavity, right? And so these bilateral symmetry organisms can either be acoelomates, pseudomates, or coelomates. And so a body cavity means that you have a gut that you can process the food in. And the number of guts that you have may vary. You may have one, you may have two. It also means that you have a coelom. A coelom is the space where you will be able to grow an organ. And so we're going to go into this here. And this is what you have to go back and remember what the formation of your derms are. So you have here, remember, the outside is the ectoderm, the middle is the mesoderm, and the inside is the endoderm. So a flatworm has one body cavity, one big entry, and that's it. So it has a tubular cavity. Acoelomates, it only has a gut, meaning it only has one entry point. It's very simple because it's only one things go one way in, and the way that it forms. Okay, this is the next one. So there's only one. So in the acoelomate, there's only one cavity. There's no space in between. So the next one is pseudo coelomate. So pseudo coelomate. So pseudo means like fake, right? And notice here that you have now a cavity here, and you have some space that is not exactly the same as a cavity in between your mesoderm and your ectoderm. So what happens is this cavity forms your gut, but now you have some space here where you can actually form um, some organs. And so roundworms will have a few more organs. They're a bit more complex, but it's still limited. You still don't have all the space that you need. The most complex organism is the coelomate. And you notice here that you have a true space, you have the ectoderm here, and you have the space here where the gut is, right? And then you have the mesoderm, and you have this whole space, this another whole space called the coelom. And this is where the organs now are going to have enough room in the mesoderm to begin to form. It's a, this would be like we would be an example of a coelomate, and we have a lot of different organs inside our body. The next so now all the coelomates will move on to the next development, and this is another challenging words, so pause your video as needed. But basically this is how embryos develop, and embryos can develop in two ways. They can develop in the protostone or deuterostone development, and I'm gonna those are complex terms. What I'm gonna do now is I'm gonna give you a lot of examples about the two so that you can understand hand in hand. So as you remember, the zygote is where the egg gets fertilized by the sperm, and then the cells begin to divide through the process of mitosis until you get to 16 cells, you get a blastula, and finally you get a gastrula, where you have the internal folding. What happens now in coelomate development is the first stage is the 4-cell development, okay? And so this side is going to be the protostone, and this side is going to be the deuterostone. So it's always going to be the protostone on this side and the deuterostone on this side. So notice that in the protostone, the 4 cells divide, and they have to have 4 cells. In the deuterostone, you can lose a cell and this will still continue to develop properly. So these are just examples of the differences between the protostone and the deuterostone, just so that you understand the difference. You don't need to memorize these. In the next section, what happens is when it goes to 8 cells, in the protostone, the 8 cells are not aligned. In the deuterostone, they are aligned. The next example is in the way that the mesoderm forms. So notice in the protostone, the mesoderm forms on the bottom. The mesoderm on the deuterostone forms at the top. And the last example, which is how essentially decides your fate as an organism, is the location of your mouth and your anus. So here, here's the coelom. In your protostone, the mouth is at the bottom and the anus is on top, and on the deuterostone is backwards. So your takeaway message is that protostones and deuterostones develop differently. You don't need to memorize these steps. I just need you to know that there's differences between them, and there's significant differences. Our last way of body plans of dividing, and now these are all protostones and deuterostones can develop in either in this way, and is the way that the body is segmented. So what does segmentation mean? It means that the body has divided into parts. So it can either be unsegmented or it can be segmented. So what does that mean? If I take this sea urchin, for example, and I cut it in half, there are no sections. All the mass, all the internal organs are all blobbed in the middle. So I essentially everything will spew out all at once. If this organism loses its pinchers, the organism will probably be okay. It only lost a pincher, right? Or you can lose a leg. So they're segmented because its organs and its complex body is divided into places, segments that